Sexual reproduction in flowering plants. Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scent and the perfume that we swoon over, the rich color that attract us, are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. They look at the diversity of the structure of the inflorescences, flowers, and floral parts, show an amazing range of adaptation to ensure formation of the end product of sexual reproduction, the fruits and seeds. In this chapter, let us understand the morphology, structure, and the process of sexual reproduction in flowering plant. Angiosperm. Flower, a fascinating organ of angiosperm. Human beings have had an intimate relationship with flowers since time of immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural value. They have always been used as symbols for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. At least 5 flowers of ornamental value that are commonly cultivated at homes and in garden. Find out the names of the 5 more flowers that are used in social and cultural celebration in your family. Have you heard the floriculture? What does it refer to? To a biologist, flowers are morphological and embryological marvels and the site of sexual reproduction. In class 11, you have read the various parts of the flower. Figure will help you recall the part of the typical flowers. Can you name the 2 parts in a flower in which 2 most important units of sexual reproduction develop? Fertilization, structure and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormone and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and female reproductive structure and endoecium and the gynoecium differentiate and develop. You would recollect that the endoecium consists of a whole of stamens representing the male reproductive organ and the gynoecium represents the female reproductive organ. Stamen, microsporidium and pollen grain. Figure shows that 2 parts of a typical stamen, the long and slender stalk called the filament and the terminal generally bilobed structure called the anther. The proximal end of the filament is attached to thalamus or the petal of the flower. The number and the length of stamen are variable in flowers of different species. If you were to collect a stamen of each from the flower, from 10 flowers, each from different species, arrange them on a slide You would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissection microscope and neat diagram would elucidate the range in shape and attachment of anther in different flowers. A typical angiosperm anther is bilobed with each lobe having two theca, that is, they are diathecious. Often a longitudinal grooves run lengthwise separating the theca. Let us understand the various types of tissues and their organization in the transverse section of an anther. The bilobed nature of an anther is very distinct in transverse section of anther. The anther is a four-sided tetragonal structure consisting of four microsporangia located at the corner, two in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grain. Structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by four wall layers: the epidermis, endothecium, middle layer, and the septum. The outer three wall layer perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the septum. It nourishes the developing pollen grain. Cell of the septum possesses dense cytoplasm and generally have more than one nucleus. Can you think of how tetrad cell could become binucleate? When the anther is young, a group of compactly arranged homogeneous cells called the sporangious tissue occupies the center of each microsporangia. Microsporogenesis. As the anther develops, the cell of the sporangious tissue undergoes meiotic division to form microspore tetrads. What would be the ploidy of the cell of the tetrad? As each cell of the sporangious tissue is capable of giving rise to a microspore tetrad. Each one is a potential pollen or microspore mother cell. The process of formation of microspore from a pollen mother cell (PMC) through meiosis is called microsporangiasis. The microspore is they are formed are arranged in a cluster of four cells. The microspore tetrad, as the anther matured and dihydrated, the microspore dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousand of microspore or pollen grains are formed that are released with the dehiscence of anther. Pollen grain. The pollen grain represent the male gametophytes. If you touch the opening anther or hibiscus or any other flower, you would find deposit of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water, take on a glass slide and observe under. a microscope, you will really be amazed at the variety of architecture, size, shape, color, design seen on the pollen grains from different species. Pollen grains are generally spherical, measuring about 25 to 30 micrometers in diameter. It has a prominent two-layered wall. The hard outer layer called the axine is made up of sporopollenin, which is one of the most resistant organic materials known. It can withstand high temperature and strong acid and alkali. No enzyme that degrades sporopollenin is so far known. Pollen grain axine has prominent apparatus called germspore, where sporopollenin is absent. Pollen grains are well preserved as fossils because of the presence of sporopollenin. The axine exhibits a fascinating array of pattern and design. Why do you think the axine should be hard? What is the function of germ pore? The inner wall of the pollen grain is called the intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grains is surrounded by a plasma membrane. When the pollen grains is mature, it contains two cells, the vegetative cell and generative cell. The vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. The generative cell is small and floated in the cytoplasm of the vegetative cell. It is spindle-shaped with dense cytoplasm and a nucleus. In over 60% of angiosperm, pollen grains are shed at this two-cell stage. In the remaining species, the generative cell divides mitotically to give rise to two male gametes before pollen grains are shed three-cell stage. Pollen grains of many species cause severe allergies and bronchial affliction in some people, often leading to chronic respiratory disorder, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminant with imported wheat has become ubiquitous in occurrence and cause pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablet as food supplement. In Western countries, a large number of pollen products in the form of tablets and syrup are available in the market. Pollen consumption has become claimed to increase the performance of athletes and race horses. When once they are shed, pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. How long do you think the pollen grains retain viability? The period for which pollen grains remain viable is highly variable and shows to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release. And in some members of Rosaceae, Legumineaceae and Solanaceae, they maintain viability for months. You may have heard of strong semen or sperm of many animals including humans for artificial insemination. It is possible to store pollen grain for a large number of species for years in liquid nitrogen -196 degrees centigrade. Such stored pollen can be used as pollen bank, similar to seed bank in crop breeding program. The pistil, megasporangium, ovule, and embryo sac. The gymnasium represent the female reproductive part of the flower. The gymnasium may consist of a single pistil monocarpellary or may have more than one pistil multicarpellary. When there are more than one, the pistil may be fused together syncarpous or may be free apocarpous. Each pistil has three parts: the stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated slender part beneath the stigma. The basal bulge part of the pistil is ovary. Inside the ovary is the ovarian cavity located. The placenta is located inside the ovarian cavity. Recall the definition and the type of placentation that you studied in arising from the placenta are megasporangia, commonly called ovule. The number of ovules in an ovary may be one wheat, paddy, mango, to many papaya, watermelon, or fish. The megasporangium ovule. Let us familiar ourselves with the structure of typical angiosperm ovule. The ovule is small structure attached to placenta by means of a stalk called funicle. The body of the ovule fuses with funicle in the region called hilum. Thus hilum represents the junction between ovule and funicle. Each ovule has one or two reproductive envelopes called interguments. The interguments encircle the nucleus except at the tip where a small opening called micropyle is organized. Opposite the micropylar end, the chalaza representing the basal part of the ovule. Enclosed within the integuments is a mass of cell called the nucleus. Cell of the nucleus have abundant reserve food material. Located in the nucleus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis. The process of formation of megaspore from the megaspore mother cell is called megaporogenesis. Ovules generally differentiate in a single megaspore mother cell in a micropylar region. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergo meiotic division. What is the importance of the MMC undergoing meiosis? Meiosis results in the production of 4 megaspore female gametophytes. In a majority of flowering plants, one of the megaspore is functional while the other 3 degenerate. Only the functional megaspore develop into female gametophyte embryo sac. This method of embryo sac formation from a single megaspore is termed monosporic development. What will be the ploidy or cell of nucleus, MNC, the functional megaspore and female gametophyte? Let us study the formation of embryo sac in a little more details. The nucleus of the functional megaspore divide mitotically to form two nuclei which move to the opposite pole, forming the two nuclei embryo sac. Two more essential meiotic nuclear division result in the formation of four nucleate and later the eight nucleate stage of the embryo sac. It is interesting to note that these meiotic divisions are strictly free nuclear, that is, nuclear division are not followed immediately by cell wall formation. After 8 nuclear stage, cell wall are laid down, leading to the organization of the typical female gametophytes or embryo sac. Observe the distribution of cells inside the embryo sac. 6 of the 8 nuclei are surrounded by cell wall and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cell within the embryo sac. The 3 cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergid and 1 egg cell. The synergid has special cellular thickening at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tube into the synergid. 3 cells are at the chalajar end and are called the antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, a typical angiosperm embryo sac at the maturity, though a nucleate is 7-celled. Pollination. In the preceding section, you have learned that the male and female gametes in flowering plants are produced in the pollen grains and embryo sac respectively, as both types of gametes are non-motile. They have to be brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grain set from the anther to stigma of a pistil is termed pollination. Flowering plants have evolved an amazing array of adaptation to achieve pollination. They make use of external agent to achieve pollination. Can you list the possible external agent? Kind of pollination. Depending on the source of pollen, pollination can be divided into 3 types. 1. Autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from the anther to the stigma of the same flower in a normal flower, which opens and exposes the anther and the stigma. Complete autogamy is rather rare. Autogamy in such flowers requires synchrony in pollen release and stigma receptivity and also the anther and the stigma should lie close to each other so that self-pollination can occur. Some plants such as Viola, common pansy, oxalis and camelina produce 2 types of flowers. Chasmogamas, flowers which are similar to flowers of the other species with exposed anther and stigma, and Cleistogamas, flowers which do not open at all. In such flowers, the anther and the stigma lie close to each other. When anther dehisce in the flower buds. Pollen grains come in the contact with the stigma to effect pollination. Thus, cleistogamous flowers are invariably autogamous as there is no chance of cross-pollination landing on the stigma. Cleistogamous flowers produce assioid seed set even in the absence of pollinators. Do you think that cleistogamous is advantageous or disadvantageous to the plant? Why? Zoetonogamy. Transfer of pollen grains from the anther to the stigma of anther flower to stigma of another flower of the same plant. Although zoetonogamy is functionally cross-pollination involving a pollinating agent, genetically it is similar to the autogamy since the pollen grains come from the same plant. 3. Genogamy. Transfer of pollen grains from anther to the stigma of different plants. This is the only type of pollination which during pollination brings genetically different types of pollen grains to the stigma. Agent of pollination. Plants use two abiotic, wind and water, and one biotic, animals agent to achieve the pollination. Majority of plants use biotic agent for pollination. Only a small portion of the plant use abiotic agent. Pollen grains coming in contact with the stigma is a change factor in both wind and water pollination. To compensate for these uncertainties and associated loss of pollen grains, the flowers produce enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by winds is more common amongst abiotic pollination. Wind pollination also requires that the pollen grains are light and non-sticky so that they can be transported in wind current. They often possess well-exposed stamen so that the pollen are easily dispersed into wind current. Figure and large, often featherly stigma to easily trap airborne pollen grains. Wind-pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. A familiar example is the corn cob. The tassel you see are nothing but the stigma and style which wave in the wind to trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plants and is limited to about 30 genera, mostly monocytolenans. As against this, you would recall that water is a regular mode of transport for male gametes among the lower plants groups such as algae, biophytes, and pteridophytes. It is believed, particularly for some biophytes and pteridophytes, that their distribution is limited because of need for water for transport of male gametes and fertilization. Some examples of water-pollinated plants are Valesneria and Hydrilla, which grow in fresh water, and several marine sea grasses such as Josteria. Not all aquatic plants use water for pollination. In a majority of aquatic plants such as water hyacinth and water lily, the flower emerges above the level of water and pollinated by insects or winds, as in most of the land plants. In Valesneria, the female flower reaches the surface of the water by long stalk and the male flower or pollen grains are released on the surface of water. They are carried passively by water current Some of them eventually reach the female flower and stigma. In another group of water pollinated plants such as Seagrasses, female flower remains submerged in water and pollen grains are released inside the water. Pollen grains in many such species are long, ribbon-like, and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water pollinated species, pollen grains are protected from wetting by mucilaginous covering. Both wind and water pollinated flowers are not very colorful, so do not produce nectar. What would be the reason for this? Majority of flowering plants use a range of animals as pollinating agents: bee, butterflies, flies, beetles, wasps, ant, moth, bird, sunbird, and hummingbirds and bats are the common pollinating agents. Among the animals, insects, particularly bees, are the dominant biotic pollinating agent. Even larger animals such as some primates, lemurs, arboreal, tree-dwelling rodents, or even reptiles, gecko, lizard, and garden lizard, have also been reported as pollinators in some species. Often, flowers of annual pollinated plants are specifically adapted for particular species of animals. Majority of insect pollinating flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small, a number of flowers are clustered into an influence to make them conspicuous. Animals are attracted to flowers by color and or fragrance. The flowers pollinated by flies and beetles secretes flow of foul odor to attract these animals. To sustain annual visit, the flower have to provide rewards to the animal. Nectar and pollen grains are the usual floral rewards. For harvesting the rewards from the flower, the annual visitor comes in contact with the anther and the stigma. The body of the animal gets a coating of pollen grains, which are generally sticky in annual pollinated flower. When the animal carrying pollen on its body comes in contact with the stigma, it brings about pollination. In some species, floral rewards are in providing safe place to lay eggs. An example is that of the latest and tallest flower of Amorphophilus. The flower itself is about 6 feet in height. A similar relationship exists between a species of moth and the plant Yucca, where both species, moth and the plant, cannot complete their life cycle without each other. The moth deposits its egg in the locule of the ovary, and the flower in turn gets pollinated by the moth. The larvae of the moth come out of the egg as the seeds start developing. Why don't you observe some flowers of the following plants or any other available to you? Cucumber, mango, pepper, coriander, papaya, onion, lavia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out which animal visit them and whether they could be pollinator. You will have to patiently observe the flowers over a few days and at different times of the day. You could also try to see whether there is any correlation in the characteristics of a flower to the animal that visit it. Carefully observe if any of the visitors come in contact with the anther and stigma, as only such visitor can bring about pollination. Many insects may consume pollen or nectar without bringing about pollination. Such floral visitors are referred to as pollen or nectar robber. You may or may not be able to identify the pollinator, but you will surely enjoy your efforts. Breeding device. Majority of flowering plants produce hermaphrodite flower, and pollen grains are likely to come in contact with the stigma of the same flower. Continued self-pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or stigma become receptive much before the release of pollen. In some other species, the anther and the stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. The third device to prevent inbreeding is self-compatibility. This is a genetic mechanism and prevents self-pollen from the same flower or other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. And third device to prevent self-pollination is the production of unisexual flower. If both male and female flowers are present on the same plant, such as castor and monoecious, it prevents autogamy but not zootonogamy. In several species such as papaya, male and female flowers are present on different plants. That each plant is either male or female dioecious. This condition prevents both autogamy and zootonogamy. Pollen-pistil interaction. Pollination does not guarantee the transfer of the right type of pollen. Compatible pollen of the same species as the stigma. Often pollen of the wrong type, either from other species or from the same plant, if it is self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen. However, it is of the right type compatible or of the wrong type incompatible. If it is of the right type, the pistil accepts the pollen and promotes post-pollination event that leads to fertilization. If the pollen is of wrong type, the pistil rejects the pollen by preventing pollen germination in the stigma or pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grains and the pistil. This dialogue is mediated by chemical component of the pollen interaction with those of the pistil. It is only in the recent years that botanists have been able to identify some of the pollen and pistil component and the interaction leading to the recognition followed by acceptance or rejection. As mentioned earlier, following compatibility pollination, the pollen grains germinate on the stigma to produce a pollen tube through one of the germ pores. The content of the pollen grains move into the pollen tube. Pollen tube grows through the tissue of the stigma and style and reach the ovary. You would recall that in some plants, pollen grains are shed at two celled condition, a vegetative cell and a generative cell. In such plant, the generative cell divides and forms the two male gametes during the growth of pollen tube in the stigma. In plant which shed pollen in the three-cell condition, pollen tube carry the two male gametes from the beginning. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enter one of the synergid through the filiform apparatus. Many recent studies have shown that filiform apparatus present at micropylar part of the synergid guides the entry of pollen tube. All these events from pollen deposition on the stigma until pollen tubes enter the ovule are together referred to as pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The knowledge gained in this area would help the plant breeder in manipulating pollen pistil interaction even in incompatible pollination to get desired hybrids. You can easily study pollen germination by dusting some pollen from flowers such as pea, chickpea, crotalaria, balsam and vinca on a glass slide containing drop of sugar solution about 10%. After about 15 to 30 minutes, observe the slide under the low power lens of the microscope. You are likely to see pollen tubes coming out of the pollen grain. As you shall learn in the chapter on plant breeding in chapter 9, a breeder is interested in crossing different species and open genera to combine desirable character to produce commercially superior varieties. Artificial hybridization is one of the major approach of the crop improvement program. In such crossing experiment, it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from the contamination from unwanted pollen. This is achieved by emasculation and bagging technique. If the female parents bear bisexual flower, removal of anther from the flower bud before the anther dehisces, using a pair of forceps is necessary. This step is referred to as emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. This process is called bagging. When the stigma of bagged flower attain receptivity, mature pollen grains collected from anther of the male parents are dusted on the stigma and the flower are bagged and the fruits allowed to develop. If the female parents produce unisexual flower, there is no need for emasculation. Female flower buds are back before the flower open. When the stigma become receptive, pollination is carried out using desired pollen and the flower bud. Double fertilization. After entering one of the synergids, the pollen tube releases the two male gametes into cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing the synergy. This results in the formation of diploid cell, the zygote. The other male gamete moves towards the two polar nuclei located in the central cell and fuses with them to produce diploid primary endosperm nucleus. PEN, primary endosperm nucleus. This involves the fusion of three haploid nuclei. It is termed triple fusion. Since two types of fusion, syngamy and diploid fusion, take place in an embryo sac, the phenomenon is termed double fertilization. An event unique to flowering plants. The central cell after triple fusion becomes the primary endosperm cell (PEC) and develops into endosperm, while the zygote develops into embryo. Post-fertilization, structure and events. Following double fertilization, events of endosperm and embryo development, maturation of ovules into seeds and ovary into fruits are collectively termed post-fertilization event. Endosperm development precedes embryo development, where the primary endosperm cell divides repeatedly and form a triploid endosperm tissue. The cells of this tissue are filled with reserved food materials and are used for nutrition of the developing embryo. In the most common type of endosperm development, the PEN undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei. And surrounding white kernel is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, pea, groundnut, beans, before seed maturation, or it may persist in mature seed, for example castor and coconut, and be used up during seed germination. Split open some seeds of castor, peas, beans, groundnuts, fruits of coconut and look for endosperm in each case. Find out whether the endosperm is persistent in cereal, wheat, rice and maize. Embryo. Embryo develops at microbial end of the embryo sac where the zygote is situated. Most zygote divide only after certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seed differ greatly, the early stage of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. Figure depicts the stage of embryogeny in a dicotyledonous embryo. The zygote gives rise to the pre-embryo and subsequently to the globular, heart-shaped and mature embryo. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledons is epicotyl, which terminate with the plumule or stem stick. The cylindrical portion below the level of cotyledons is hypocotyl that terminate with layer in the radicle or root tip. The root tip is covered with the root cap. Embryos of monocotyledons possess only one cotyledon. In the grass family, the cotyledon is called scutellum, that is situated towards one side lateral of the embryonal axis. At its lower end, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath called coleregia. The portion of the embryonal axis above the level of attachment of scutellum is the epicotyl. Epicotyl has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure. The coleoptile soak a few seed of water, say of wheat, maize, peas, chickpeas, groundnut overnight, then split the seed and observe the various parts of the embryo and the seed. Seed. In angiosperm, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Seed are formed inside fruits. The seed typically consists of seed coats, cotyledons, and an embryo axis. The cotyledons of the embryo are simple structure, generally thick and swollen due to storage of food reserve as in legume. Mature seed may be non-albuminous or ex-albuminous. Non-albuminous seed have no residual endosperm as it is completely consumed during embryo development. P. groundnut. Albuminous seed retain a part of endosperm as it is not completely used up during embryo development. Wheat, maize, barley, castor. Occasionally in some seeds such as black pepper and beet, remnant of nucleus are also persistent. This residual persistent nucleus is the perisperm. Integument of ovule hardened as tough protective seed coats. The micropyle remain as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. As the seed matures, its water content is reduced and seeds become negatively dry. 10 to 15% moisture by mass. The general metabolic activity of the embryo slow down. The embryo may enter a state of inactivity called dormancy, or if favorable conditions are available, adequate moisture, oxygen and suitable temperature, they germinate. As ovules mature into seeds, the ovary develops into fruits. That is, transformation of ovule into seed and ovary into fruit proceed simultaneously. The wall of ovary develops into wall of fruits called pericarp. The fruits may be fleshy as in guava, orange, mango, etc., or may be dry as groundnut and mustard, etc. Many fruits have evolved mechanism for dispersal of seeds. Recall the classification of fruits and their dispersal mechanism that you have studied in an earlier class. Is there any relationship between number of ovules in any ovary and the number of seeds present in a fruit? In most plants, by time the fruit develops from the ovary, other floral buds degenerate and fall off. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contributes to fruit formation. Such fruits are called false fruit. Most fruits, however, develop only from the ovary and called true fruit. Although in most of the species, fruits are the result of fertilization, there are few species in which fruits develop without fertilization. Such fruits are called parthenocarpic fruits. Banana is one of such examples. Parthenocarpic can be induced through the application of growth hormones, and such fruits are seedless. Seeds offer several advantages to the angiosperm. Firstly, seeds' reproductive process such as pollination and fertilization are independent of water. Seed formation is more dependable. Also, seeds have better adaptive strategy for dispersal of new habitat and help the species to colonize in other areas. As they have sufficient food reserve, young seedlings are nourished until they are capable of photosynthesis of their own. The hard seed coat provides protection to the young embryo. Being product of the sexual reproduction, they generate new genetic combination leading to variation. Seed is the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for storage of seeds, which can be used as food throughout the year and also to raise crop in next season. Can you imagine agriculture in absence of seeds, or in presence of seeds which germinate straight away soon after formation and cannot be stored? How long do the seeds remain alive after they are dispersed? This period again varies greatly. In a few species, the seeds lose viability within a few minutes. Seeds of large number of species live for several years. Some seeds can remain alive for hundreds of years. There are several records of very old yet viable seeds. The oldest is that the Lupinus arcticus, excavated from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. A recent record of 2,000 years old viable seeds is of the date palm, Phoenix dactylifera, discovered during the archaeological excavation at King Herod's place near the Dead Sea. After completing a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproduction capacity of some flowering plants by asking the following questions. How many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? how many flowers are present in a tree, and so on. Can you think of some plants in which fruits contain very large number of seeds? Orchid fruits are one such category, and each fruit contains thousands of tiny seeds. Similar is the case in fruits of some parasitic species such as Orange and Stigma. Have you seen a tiny seeds of ficus? How large is the tree of ficus developed from that tiny seed? How many billions of seeds does each ficus tree produce? Can you imagine any other example in which such a tiny structure can produce such a large biomass over the years? Apomix and polyembryony. Although seeds in general are the product of fertilization, a few flowering plants such as some species as Asteraceae and grasses have evolved a special mechanism to produce seeds without fertilization called apomixis. What is fruit production without fertilization called? Thus, apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction, division, and develops into the embryo without fertilization. More often, as many citrus and mango, there is some of the nuclear cells surrounding the embryo sac start dividing, protrude into embryo sac, and develop into embryos. In such species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred as polyembryony. Take out some seeds of orange and squeeze them. Observe the many embryos of the different size and shapes from each seed. Count the number of embryos in each seed. What would be the genetic nature of apomictic embryos? Can they be called clones? Hybrid varieties of several of our food and vegetable crops are being extensively cultivated. Cultivation of hybrids have tremendously increased productivity. One of the problems of hybrids is that hybrid seeds have to be produced every year. If the seeds collected from hybrids are sown, the plants in the progeny will segregate and do not maintain hybrid character. Production of hybrid seeds is costly, and hence the cost of hybrid seeds become too expensive for the farmer. If these hybrids are made into apomix, there is no segregation of character in the hybrid progeny. Then the farmer can keep on using hybrid seeds to raise new crops year after year, and he does not have to buy hybrid seeds every year. Because of the importance of apomixis in hybrid seeds industry, active research is going on in many laboratories around the world to understand the genetics of apomixis and to transfer apomictic genes into hybrid varieties.